year reports. Therefore, it is not simple to cover in 2 hours. That's why I divided into two sessions. One is the part one. In this part one, see, we will be covering the sample collection, transportation, and then analysis and interpretation of data will come later on. Because from the sample collection till the transportation, it reaches the laboratory is the prime importance. See the sample collection. Let's start from the sample collection. सब लोग सुना है ना मेरा आवाज आ रहा है ना सबको okay so let us start from the sample collection. First we have to see what is sample. We are telling sample collection. So what we have to collect, what is meant by the sample? See the sample which is written there, you can read it. A sample is a portion of material. That means we cannot take the whole material. So it is a portion of material. And it should be small enough because we have to carry. So it is a portion of the material small enough in volume to be transported. But even though it is to be small enough, it should be convenient for transport, but yet large enough to analytical purposes. For analytical purposes, it should be a minimum volume we should take it. If it is a water sample, what should be the volume? It depends on the number of samples, the number of parameters that we have to analyze it. That means minimum 1 liter or so, minimum 2 liter sample may be required usually. But suppose toxicity, we are asking toxicity, they have to test for toxicity. It may require 15 liter of water sample. So the sample means an actual representation of the material, small enough for convenience of transport, but large enough for the analytic purposes. Then another more important is next one, that is while still accurately representing the material being sampled. We have to ensure that the sample collected is actually representing the material to be sampled. Why it is so? For example, I will tell you. Suppose we are drinking water. We are storing drinking water in our home. Let us go to villages, normal villages, and they will be having some utensils there. Some pot will be there. Some people may be using the aluminium pot, or some people may be using the pot made of this mud or something. So in that case, suppose they are putting water sample for drinking purposes. When they are collecting the water sample for drinking purposes and keeping at home, it is safe. Then how they are taking this water for drinking? Suppose they are using a cup, immersing in the pot and taking water. Suppose if the cup is not clear, or if the cup is not clean enough, or the cup is somewhat contaminated, or the cup is somewhat ugly, then when they are dipping into the pot of the pot in which drinking water is kept, then the water will get contaminated. That means the water they are drinking may not be the actual water which is stored in the pots. Just like here, the main thing is that the sample collected should be representing the actual material. So, what is there in the material that is to be collected? So, if the container that we are taking the sample, if it is not good enough, or if it is dirty, or if it is contaminated, then taking the sample into this container will not give actual representative sample. So the thing is that we have to collect the sample in its actual representative. Actual representative, how it comes, you will see. Then see, the relative proportions of concentrations of all pertinent components will be the same in the samples as in the material being sampled. So we have to ensure that the sample that we are collecting represents the actual material. Suppose there is a flowing water. Flowing water may contain sometimes suspended solids, or sometimes the floatable solids, or sometimes the dissolved solids, or it may vary from time to time. If it is so, how we have to collect the sample? If there is suspended solid, we have to ensure that continuously the suspended solids is also collected. Otherwise, what will happen? If you are collecting the sample, you are flowing water sample, keeping it in a tank, and then collect the supernatant sample, it will not give you the representative thing of the material. So the sample, what is sample? Sample is a portion of material, small enough for the transportation convenience, but large enough for the requirement of analysis, but still representing the actual material. That is important. Then second thing. So first thing is sampling. We ensure that we have done the sampling accurately. Second thing is the transport. Because we are doing analysis in the laboratory. So in the laboratory, we have to ensure that the actual representative sample we have collected. And further, we have to ensure that actual representative sample, whatever we have collected, is the sample that reaches the laboratory. That means in between the collection and the transportation and the reaching the laboratory, the material should not change. The concentration of the components or anything should not change. There we have to ensure that the transport, what is transport? The sample will be handled in such a way that no significant changes in composition occur before the tests are being made. That is significant changes. What is significant? See, suppose we are collecting water sample from a muddy area, or water sample from a place where it is decayed materials are there, or eutrophication has happened, or the sulfide content is there. Suppose we are collecting the sample, then transporting it to the laboratory, the sulfide content will immediately change. So we should ensure that it is transported in such a way that the components in the sample is not significantly changed. So two things are important: sample collection and secondly is important. So first we have to know what is sample. Unless and until we do not know what is the sample. So a sample is a representative one of the material. Representative one means when we are collecting sample, the morning sample may not be same of the evening sample. So if we are collecting samples in 3 occasions, same time may likely to be used, whatever it is. Okay, so I think now we are familiar with the two terms. One is the sample and the second is the transportation. And the importance of sampling and the importance of transportation. Am I right? Am I right? I think you understood these 2 points. No response from anybody? Can you hear?
Yes, sir.
Okay. No response means it can be everything understood or it can be nothing understood. Next. So the precision and accuracy. Precision and accuracy are the two terms that is usually employed in terms of analysis. What is the precision and accuracy of the result? How precise and how accurate the result is, that is what the baseline data gives. The baseline data will be precise and accurate only if this laboratory gets the sample correctly. That means the precision and accuracy of the test result will depend upon the material the laboratory has received. So the first point is the person who is collecting sample, the person ensuring that the representative sample is collected, person ensuring that the representative sample reaches the laboratory. Then the first thing let's rest with the laboratory paper. So the precision and accuracy of the result is dependent on we are depending EIA paper, we are preparing EIA report, we are depending on the accurate results. So that is sample who is collecting and who is transporting, and if the laboratory receives the correct representative sample, then the analysis will and result will be accurate. Then analysis also depends on, interpretation also depends on. So first thing is the sampling and transportation. Then see, so the precision and accuracy of the result of any test on the quality of environment is the resultant of all the efforts that lead to the final result. So when EIA is prepared, we are taking the final result, that is the result analysis report we are taking. So the final result, what is reported by the laboratory, we are taking. But that result is dependent on first one, collection of sample, that is a representative sample. Second one is the handling and the chemical treatment. And next is the method of storage. And another is the chemical analysis and calculation interpretation of the data. Now in this session, I am not covering the chemical analysis and calculation interpretation of data. That will be covered in the chapter, this is a part 2. See, if about all these things, all these steps, if these steps are carried out with insufficient care, that means these steps are to be carried out with a sufficient care. They should be carried out with very, very careful attendance is required for this. So if any of these steps are carried out with insufficient care, the final result will be only a number. It will not give the actual condition of the environment. So the concentration of a given component will be no more than a figure without relation to the actual situation of the environment. So if these things, if any of these things go wrong, then the baseline data will also be wrong. It starts from the sampling. Suppose the person who is transporting, or the person who is analyzing, or the person who is interpreting the data, but the sampling has gone wrong, that means everything is gone. So starting from the sampling, it is very important. That is the importance of sampling and transportation. Let us see sampling water. We have to do the sampling of water. What are the things that we have to take care? What is the site selection? We have to be careful while selecting the site from where we have to collect the sample. Whether it is a water body or water, or the stagnant water body, or the flowing water body, or it is a open well, or it is a bore well, whatever it is. From where we have to select the sample. So the prime importance is site selection. Site selection means from which point I have to take the sample. What will be the, where can I get a representative sample, and where I have to collect the sample? See, so the site selection is the first thing. Second thing, what is a checklist for sampling and the sample labeling. Suppose you are going for sample collection and they are collecting 4 or 5 or sometimes n numbers of samples. Then noting it in the notebook will not do. We have to have checklist for sampling, whatever things we have to take it, whether the plastic cans you have to take it, or these hop wires you have to take it, or the bottles you have to take it. You should have a checklist. The sampling points. You have selected sampling points, and from there you have to take the sample. So you have to have the checklist, whatever things you have to take to the site. And you should also have the what is the items for sample labeling. Each sample to be labeled. You have to say from which location you have taken. If you are giving a code number also, that you have to make it. And the type of collection, date of collection, and atmospheric conditions you have to say. You have to note it. So sample labeling. That is what the time of sample collection. Because if it is a water sample, say morning sample, if you are taking morning water sample, the temperature will be less than the atmospheric temperature. But in the afternoon, if you are taking, the water temperature will be a little more higher than the atmospheric temperature. So the time is also important, because when you go for the BOD analysis, it also is somewhat dependent on the temperature, water temperature. That is why earlier, even though we were doing the BOD 5 days at 20 degrees centigrade, in our country it is not the normal temperature, so it is changed with the room temperature 27 degrees centigrade. So acclimatize with the normal conditions of the water bodies in India. Then see. Then you have to decide on the type of sample. What is the type of sample? Graph samples are there, composite samples are there, integrate samples are there. What is graph sample? You take sample from one point, that is enough. You think that it is representative. Then what is composite sample? Suppose there is a lake. Like you are taking one sample will not be opportunity. You are thinking that, okay, I will take some 10 square meter area, and from the 10 square meter area, all these points may not be having the same composition. So I think that I may take one from the north side, and I will take from the central and the east or west or this much. This will divides into 10 square meter. I am taking from 4 to 5 points. From each point, I am collecting 1 liter sample. Then I am mixing it. See, from one point you are taking half a liter sample, another point 1 liter, another point 1.5. It should not do. You have to take the same volume, 1 liter, 1 liter, 1 liter, 1 liter, or half liter, whatever it is. That you can decide. Then mixing all these together and taking some 1 liter or 2 liter, what is the requirement? Taking that. That is a composite sample. Then another is integrate. What is integrate sample? See, in some industries, you can see that you want to test for the efficiency of the ETP, or you want to test the efficiency of the STP. See, the inlet water that is coming towards the treatment plant may not be having the same composition at all times. Suppose early morning time it may be having some contaminants, and at that time after 10 o'clock or 11 o'clock or 12 o'clock or 1 o'clock, then evening or night time, it may be different. But you want to see, then you want to see what to do. You will take what samples, decided volume. Earlier I have told 1 liter, 1 liter, 1 liter, just like in the early morning you want to take that 10 o'clock or 5 o'clock or 11 o'clock or in the night time to mix that many times. Take equal volume of samples, then mix it and give for analysis. So that will form the integrated sample. So you have to decide what type of sample you have to collect, whether it is gram sample is enough, composite sample or integration. Suppose you have well water, you are collecting that gram sample is enough. So you have to decide from where you are collecting, the nature of the source, and then you have to decide gram sample or composite or integrated. So when you prepare the checklist for sampling and sample labeling, all these things should be there in your minds. Otherwise, you will not be carrying in a sufficient sufficient materials for you to help in analysis, help in selecting sample. So you have to think of type of sample you have to collect: gram, composite, or integrate. Then another one, type of containers. You cannot collect all these samples in plastic cans. Some samples can be samples can be collected, or samples of certain parameter analysis can be collected in glass bottles only. Some cannot be collected in glass. Some may require polyethylene or Teflon. So you have to decide of the type of container you have where you have to collect sample before going to the site for sample selection. See, in the case of mercury and orthophosphate, only glass can be used. But if you want to analyze the boron, you cannot use it. You cannot collect the sample in borosilicate glass. You could not use the glass bottles. There you may require the polyethylene or Teflon bottles. Then another thing, there are in situ measurements also. In situ measurements, you have to carry instruments and reagent solutions. There are certain parameters that is to be done in the site itself. See, when functional area experts are going to monitor how the monitoring people are doing, how the sampling they are doing, how this monitoring work is going on, they have to see whether these people has carried the instruments required for this sampling, and they have the instruments that whatever required for the in situ measurements. See, pH can be done, pH can be measured at the site, or if you want the conductivity also, it can be measured at site. And when it is sent to laboratory, if the conductivity drastically changes, you can compare it why it has happened. Then some reagent solutions are also required. That reagent solutions what are required come later on. Why it is told like that? Then how to collect the sample? Rinse the container 3 times with the sample. Suppose you are collecting sample in a plastic can. You will wash it. No, if it is a new one, okay, you will wash it. But again, you have to rinse the container 3 times with the sample you are going to collect. If it is river water, you take the water, wash it, and clean it. And you repeat it 3 times. That means even if it is there, suppose the can may have some water in here, you have used it, washed it. So if you rinse it 3 times with the sample you are going to collect, that means it will ensure that the container will give you the representative sample and no dilution happens or no chemical reaction happens. Then another thing what is to be taken care, type of filling the container. कुछ लोग पूछेगा अरे ये क्या है ये bottle I have to fill it fully or I have to take it half fully. Yes, if you are going for the chemical analysis, you fill it fully. If it is for the bacterial analysis, don't fill it fully. This is to be known. That is, for what purpose you are collecting the sample and for what are the parameters that you are expected to analyze from this sample in this container, you have to know. By that, in that way, you have to think or decide of the type of filling the container. Okay? Is it okay? I think you understood anything or you didn't understand anything? Anybody can answer? Can you hear?
Yes, sir.
Yes.
You are understanding or not? कुछ समझता है या नहीं समझता है?
समझ में आ रहा है? समझ में आ रहा है?
आवाज आ रही है?
आवाज नहीं आ रही।
You are hearing something or you understanding something? कुछ समझता है या नहीं समझता है?
कुछ समझते आवाज कम कर।
मेरा आवाज आ रहा है क्या? आप लोग कुछ समझता है या नहीं?
So they are saying some of what
Okay.
you are saying is understandable, some of it is not because of maybe the voice issue in the system.
आवाज कम करेंगे।
आवाज आ रही है?
Yes, sir. Continue, sir.
Okay. Shall I continue?
Yes, sir.
Yes, sir.
Okay. Then come to the surface water samples. Now, water sample means it is many types of water samples are there. Let us see the surface water sample. A lot many people will have confusion from where we have to collect it and what depth we have to collect it. See, in the case of surface water sampling, samples will be collected from well mixed section of the river. Usually we think of collecting river sample. From where we have to collect well mixed section. That means if you go to the river and collect from the sides of the river, there will be stagnation. So a person who is going to collect the water sample, you can observe whether there is stagnation or it is simply flowing. So you are a person who is going to collect the sample, you can see where you feel that there is a regular mixing of water. From there only you collect some sample. So well mixed portion of the river. Where 30 centimeter below the water surface. 30 centimeter below the water surface means you need not go take a scale and go for 30 centimeter approximately. That means don't collect water from the surface. You have to go to a little depth. अरे ये कैसे हो सकता है? Say 30 centimeter below the water surface using a weighted bottle or your sampler. Sampler we will have. You know the sampler, putting a messenger, it will take the water from that place only, from the 30 centimeter only. I'm not showing that instrument, so many instruments are there, water sampler. Or if you do not have any water sampler, simply you can take a bottle, put some weight below it, and have the lid in it. And lid also you have to tie with another strip. Then take this bottle, put the bottle inside, it will go down. You are thinking that when it has gone up to some 30 centimeter, you take the lid. Water will be built from that region. Even if water sampler, costly water sample is not with you, you can ensure that you have collected water nearly about 30 centimeter depth in this way. Simple system. Then second, reservoir samples you have to collect, and from the canals also we have to collect samples. Samples from reservoir sites will be collected from the outgoing canal. So if a reservoir is having canals, or there is for irrigation purposes or something, from the canal you have to collect it. Where? From the canal, power channel, or water index extraction in case water is pumped. See, if water is pumped, pumping station is there for water supply, municipal water supply, some pumping station is there. So near to the water pumping station, in the water intake place, you can take it. Usually some days it may be a gravity well. If it is a gravity well, from the water intake point you take it, or from the canal, that is the outgoing canal. Then there will be a question, if the canal is not having any water, because there are some seasonal discharge is only taking place from that reservoir to the canal if it is so. That's another point. When there is no discharge in the canal, sample will be collected from the upstream side of the regulator structure. That means where the water intake is there, from this upstream only you take. The upstream is not so far, near to the upstream you take the sample. Then next is very important. The DO in the sample must be fixed immediately after collection using chemical reagents. DO means dissolved oxygen. I have seen sample people collecting samples for dissolved oxygen analysis and keeping the bottle and sending in the water. But it should not be done because the dissolved oxygen content may change when you collect the sample and send it to the laboratory. And on the way of transportation itself, the dissolved oxygen content may change. So, at the point of sample collection, itself you have to fix the dissolved oxygen. Before that, you have to be careful that dissolved oxygen content is not changed by you collecting the sample. See, taking a mug or taking a bucket, collecting water and put the mug and put in the bottle will not do. The dissolved oxygen content will change because it will cause bubbling. If bubbling is caused during the sample for the dissolved oxygen, then the DO will change. So the sampling should be made through a siphon. It is to be a siphoning method only you have to collect. If you do not have the siphoning method, simply you take a small tube. Small tube where you are using in the, what is it, in aquarium pump you are using very small tube. No? You take a tube, put it in and drag water, then put it. So it will make a siphon. This is a simple siphon. So by siphoning, without bubbling only, you have to collect for the dissolved oxygen in a bottles for the dissolved oxygen and any sample. Then immediately after the collecting the dissolved oxygen sample for dissolved oxygen by siphoning method, it must be fixed immediately after collection. What is fixing? That is dissolved oxygen content, you have to fix it. How you will fix it? You have to fix it by adding some chemicals. What is a chemical? One is the manganese sulfate solution. Then second is the alkali iodide acid. Because the method is called the Winkler modified method. That's the analysis. So first you have to add manganese sulfate solution, 2 ml manganese sulfate solution. And after that you have to add 2 ml iodide acid solution, alkali iodide acid solution, if it is a 300 ml bottle. So if the water is collected for analysis of dissolved oxygen by siphoning into a 300 ml bottle, first you add 2 ml manganese sulfate solution and then 2 ml alkali iodide acid. At the time of adding these two also you have to ensure that no bubble is going in. That means you have to take the pipette and put it by nearly about a half of the bottle, then only release to your well. If you put it over the bottle, then it will cause bubbling, small bubble. It will also change your result. Then by adding these manganese sulfate and iron acid, you will get a brown color. You shake it well. You shake it well. Once it is shaken and you see that sometimes if it becomes a white color, that means there is no dissolved oxygen. From the color, see the color itself, we can say how much will be the dissolved oxygen. It is highly brown, then you can say that it is having high dissolved oxygen. Then after that, this dissolved oxygen, you have fixed the dissolved oxygen, then shake it well and keep it. It can be sent to the laboratory. While reaching the laboratory, we can see that there is a precipitate inside the bottle, settled down. That is the dissolved oxygen. Then by the back titration, they will do the analysis. That analysis part now I am not coming. Next session, next part I am coming. So in the field, you have to fix the dissolved oxygen content. That is, dissolved oxygen content is already fixed. Manganese sulfate and iron acid, alkali iron acid, you have added, and it is changed into manganic state. Then in the laboratory, you will change another reaction. So manganic state oxygen is captured by this manganese. Then next, groundwater sample. In groundwater sample, open duct wells is used for domestic irrigation water supply. You can use it for groundwater sampling. Open duct wells, usually that we are doing. Tube wells fitted with a hand pump or a power-driven pump for domestic water supply irrigation. Tube wells are having the pump. You can put it. You can take water from there also. But there are points that you have to be careful. That piezometer is purpose built for recording of water level and water content monitoring. From there also you can collect a groundwater sample. But see, open duct wells which are not in use or have been abandoned will not be considered for water quality monitoring. But it can be considered for water level monitoring. So be careful in this. Open duct wells which are not in use. Suppose you are going to an area and asking people whether this is an open well. Okay, you can take sample, but you have to ask them whether it is put in use. Suppose they are telling it is not put in use for so many years, then don't take water for quality purpose analysis. You can take water for, you can use that well for water level monitoring. Got it? So this point should be very careful. Then groundwater sample. Continuing with the groundwater sample, see, use the weighted water sample to collect sample from an open well about 30 centimeter below the surface of water. Usually, what when they open well, what people do, usually put a bucket and take water and take it. But they are also weighted bottle. Earlier I have told from the river. If you don't have the water sampler, simple water thing you have put it, weighted bottle. Do not use a plastic bucket which is likely to skim the surface layer only. You will take the plastic, this bucket, only the surface water you will get it. See, in some open wells you can see that the upper layer is having some some colors, just like the rainbow colors. Some rainbow colors you will see in the outer surface layer of the open wells. Why it is so? Because it is having the iron bacteria. Because of the action of the iron bacteria, this rainbow colors has come. This water may be having iron content also little more. If you collect the water sample from that well using a plastic bucket, you will get the surface water sample only. Then iron content will be high. Your result will be wrong. So you have to collect water from little below surface, because in the upper layer it will be the iron content will be in ferric state, ferric FeRic. But in the lower layer it will be ferrous state, it will be a green color. So it will not give the repository sample. Therefore, use the weighted sample in open well also. Then samples from the production tube wells to be collected after running the well for about 5 minutes. If you are collecting sample from the tube wells, you allow the water to run for 5 minutes, so it will go. ऐसे जाने के बाद you take sample. Then non-collection piezometers can be purged using a submarine pump, but the purged water volume should be equal to 4 to 5 times the standing water volume before sample is collected. You have to have an approximate calculation of the water quality. No, quantity. 4 to 5 times you pump it off, then collect it. Another one, when for bacteriological purposes, when samples collected from tube wells or hand pump, the spout or outlet of the pump should be sterilized under flame. So when you are going to a site for collecting water samples, for biological purposes also, either you have to have a spirit lamp or at least you have to have a candle and a matchbox. Why? Because the tip of that tap should be heated so that the contamination is not there. So it has to be heated for some time, then allow the water to flow for some time, then collect the sample. Okay, so this is very careful when going for the collecting the samples for bacteriological analysis. Then observation. Once the samples are collected, then you are sending it for analysis to the laboratory. So it should be preserved. That earlier I have told sample. What is sample? I have told. Representative 1. And what is transportation? There should not be any change in the characteristics of the sample you are to ensure it. So you have to do some preservation also. A lot many preservation methods are there. You can see in the standard methods for examination of water and wastewater of the APHA. American APHA 23rd or 24th edition has come. You will see a lot of preservation methods, but sometimes it may not be practicable. At least do the practicable things that only I'm telling. So many are there, but at least do the practicable ones. That is, for BOD and the bacterial analysis, the sample should be kept at a temperature below 4 degrees centigrade and in the dark. So, for that, you can take an ice box. So, while going for sampling itself, you should have the ice box, and inside should be have the ice cubes also. So, for BOD, etc., you are taking sample of the biological samples. Immediately after taking the sample, you keep it in this ice box. Then for COD. COD, if the sample is taken and on the same day the analysis cannot be done, usually it is done after 24 hours only because the transportation may take some time or by within 48 hours only. So for the COD, you have to, what you have to preserve below pH 2. You have to ensure that it is below pH 2, sample is preserved below pH 2. How will you test the pH? You have to have the litmus paper with you. Add this sulfuric acid, concentrated sulfuric acid, then mix the sample, then test of this with using a litmus paper. So while going for the sampling site, you should have litmus paper also with you. Yes, that is a checklist I have told first. So if the COD for analysis, you for the COD, they take the sample, preserve the sample below pH 2 by using sulfuric acid, concentrated sulfuric acid. In the case of ammonium nitrate, total oxidized nitrate, that is a total gel nitrate TKN, and the phenol analysis, these same procedures to be followed, that is to be preserved for below pH 2 using concentrated sulfuric acid. Then another thing is there also pH is below 2, but it is to be preserved using preserved using concentrated nitric acid. So here I have told for the COD, ammonium nitrate, TKN and phenol analysis, you have to make it below pH 2 using concentrated sulfuric acid. But in the case of metal analysis, those two samples to be brought to below pH 2 with the concentrated nitric acid. If it is preserved with the concentrated nitric acid at pH below pH 2, even 6 months also it will be stable. The metallic content will not change. But if it is a mercury, in the case of mercury, it should be carried out within 1 month. Let us say 4 to 5 weeks. Because mercury analysis and other is different. In the case of other metals, you can use the cathode method of the AIS, atomic absorption spectrometry method. But for the mercury determination, it is a cold vapor technique. It is different from the atomic absorption. So other methods are done by the cathode, that is the atomic absorption method, or another method, anything. But mercury determination is mainly done by the cold vapor technique using mercury analyzer. So it should be analyzed within 1 month or 5 weeks. Then transported to concerned laboratory, preferably within 48 hours. So next, within 2 days, it should reach the laboratory. Then coming to the air sample. Air sampling. See, the water sample, I have told, I think now we can come to the air sample. See, the photograph you have seen, the water sample. The credit of these photographs goes to somebody else. The water sample, this water area sample credit goes to Regis. And this air sample photographs, the credit goes to, I think, Siddharth. So these are only natural photos. I have not taken these photos from the net. Natural photographs. See, collecting air or emission to identify and quantify the contaminants. That is the air sample. One, collecting air, that means ambient air. Second is the emission from the stack air. For what? To identify and quantify the contaminants. What are the contaminants? What are the contaminants? And how far are the contaminants? So quantify and quantify. In air sampling, we have to think of the non-point sources and point sources. Non-point sources we cannot say. In atmosphere, we cannot say that. But point sources, it is stack emission usually. Usually stack emission. Of the automobile exhaust is also a point source. In ambient air, what are the things we are measuring? Sulfur dioxide, oxides of nitrogen, PM10, PM2.5, carbon monoxide, ozone and hydrocarbons. These are the usual things. So many other things are also there. In stack monitoring, that is on the stack we usually mention sulfur dioxide, oxides of nitrogen and particulate matter. How we are reporting? We are reporting in microgram per meter cube or microgram per nm cube, except the carbon monoxide, which is reported in milligram per meter cube. See, why I have told this? Here you see, we are taking the analysis report that we receive from the laboratory and we are giving interpretations. You have to see whether the parameters are reported in microgram per m cube. If it is stack emission, they have to report the microgram per nm cube only, not m cube. Because it has to be what is the standard conditions of temperature and pressure. If the stack emission, if the emission details are given and the stack emission parameters are reported or the emissions are reported, it cannot be microgram or m cube because the stack temperature differs. There the calculation, the laboratory people has to do the calculation for the standard temperature and pressure. There they can report in the microgram or nm cube only. So that's the difference. But in carbon monoxide, you see, it is not microgram, it is milligram or m cube. See, I have seen laboratory reports that we have received, where the carbon monoxide is also reported in microgram per m cube. You should know, you should ask the laboratory people, hey, kaise, ye carbon monoxide report kaisa microgram per m cube aaya, it is milligram per m cube. And also time weighted average, 24 hours for all except 8 hours for carbon monoxide and ozone. So carbon monoxide and ozone time weighted average is 8 hours, it is not 24 hours. So when you see the report also, when you are going through the modeling also, when you are interpreting the results also, you have to ensure that these are like that only. That is a somewhat complicated case in the air sampling. You should be very careful. Then, selecting monitoring locations. It is very important. In the case of water also, I have told how you have to select the monitoring stations now. Just like the ambient air also, you should be careful in selecting monitoring stations. See, when we are preparing the AA, and we are also outsourcing this one, outsourcing these to some parties. We may be giving these water sampling stations and air sampling stations or the monitoring points we will be giving. But I have seen that in air monitoring stations, usually what we say that downstream wind, upstream wind direction, crosswind direction. See, this is not enough. While selecting the water monitoring station, by selecting monitoring stations for the water sample, it is very simple, very simple. It's very simple. But for selecting air quality monitoring station, it's not very simple. Simply upstream, downstream, and upwind, downwind, crosswind will not do. See, what are the things you have to keep in mind. See, one major existing air pollution sources are potential nearby source. You see near to your industry or near to your proponent, near to your proposed project, is there any major industry air pollution source nearby? Because it is likely to cause cumulative pollution in this area also. If it is so, you put a station nearby to it. Then only you can say the cumulative effect, otherwise you cannot give an interpretation of the cumulative effect. And you should be knowing why you put this station, because by selecting the station itself, you are thinking that while going for the interpretation of the data on the cumulative effect of this one, I need this point. You look at this point and see the your cursor. Second, topography features of the site. Air pollution, see, air should, when we think of the watershed also, there is air city is also very important. How the terrain is there. If your proposed project is lying in a valley area and two hills are there on both sides, what will happen? See, it is likely to have inversion for so many times, not for the particular time only. Majority of the time it will having inversion only. Suppose there is a hilly area one side and another side there is no hilly area. Then what will happen? There in from this industry, if they are having this tag, there are majority of chances to have heat islands to be produced in the hilly region, not in this valley area. So considering this, you have to take the mountain stages. So terrain, then lakes, river valleys, coastline, etc. These things you have to take care. Then meteorological conditions. Meteorological conditions you have to see. What is the season of that period? Or what happens during the change of the seasons? Whether there is the extreme summer season or extreme winter season. You have to think of this. In extreme winter season, what will happen? What is the frequency of this inversion in that area? You have to think of it. There are likely chances that at a particular point, and frequently inversion happens. If it is so, you have to put a monitoring station at that point also. That is very important. That's a meteorological condition. Then population distribution. If it is a highly populated area, or the population is very higher, that point also needs to be put as a monitoring station. See, population, when I say, don't think that it is the human population only, it can also be wildlife. If there is wildlife population also, densely populated wildlife is also there, that is also important. Suppose even the monkey concentration, monkeys are inhabiting a particular area with a high concentration, high population, that area is also very important. So not only human population, if human population is there, then the highly dense area of human population distribution, you see. If it is a hospital area, if it is a primary school area, if it is an area where the old age homes are there, these are very important. So those areas should also be taken for the monitoring location. That's why I'm telling. When we take the monitoring location for air quality monitoring, we simply say that upwind, downwind, crosswind, finished. No. These are very important. Then major roadways and areas of traffic congestion. Major roadways and areas of traffic congestion, I am telling, it is to be considered in your mind for not for selecting monitoring station, but for not selecting monitoring station. Because I know near to the traffic point, I will get a high sulfur dioxide or high NOx, I will get it, or high SPM, I will get it. So why should I take that one? I am interested in other areas only. In daycare schools are there, children's schools are there, LP schools are there, hospitals are there, old age homes are there. Those are the things I am interested. So consider major roadways and areas of traffic congestion. Don't take that area, need not take it. Then land use pattern, whether it is used for the agriculture purposes, or it is as a barren land, or it is for the aquaculture. So these things, land use, you have to think of it. Whether the monitoring location should be kept there, because if it is agricultural land is being surrounded to your proposed project, and this proposed project is a cement project, or it is a chemical fertilizer plant, then what are the emissions that is likely to come out from here? It is the NOx or this other chemicals also which is coming out, just like the cement, it is a dust. Cement dust, somewhat cement dust, if it reaches the leaves of certain plants, it will grow. But cement dust, if it reaches the plants, if it reaches the stomata or some other plants, its growth will be retarded. But some vegetables it is good, some vegetables it is not good. That is why you have to think of the land use pattern also while selecting monitoring location. And you have to think of the proposed project, the activity of proposed category of project also for selecting this one. Then second, another one, building structures that influence building downwards. What is the building structure and building downwards? So the air is passing from one place, then building structures it is preventing it. So the air will come out, then go downstream. So it is likely to cause another way of heat island effect. That is why you have to think of the building structures that influence building downwards. Very important. Then existing ambient air quality monitoring network location, if any. If a national ambient air quality monitoring location is there at that point, why should you put another point? Not required. You can take it from the NACM data. Then representative of site specific or nearby EMD station. If you have nearby EMD station for you, put your monitoring station there. You could take another points. Any nearby ecologically sensitive area. If ecologically sensitive area there, that also you have to think of it. It is very sensitive. Then nearby non-attainment areas. Non-attainment areas means critically polluted or severely polluted or legally specified areas. Legally, it is told that it is a very critical area. These defense installations are there, or the missile projects are there. So, or some defense of something like that is very sensitive to the security of the country is there. That is also non-attainment. It is specifically mentioned. Critically polluted area is also there. Severely polluted area also will come on that non-attainment areas there. If it is so, you should be very careful in putting monitoring stations. Okay. So these are the points while selecting monitoring locations for the air sampling. So keep in mind, it's not easy for selection of air sampling locations, as is the case of water sampling. So it is very complicated, and you have to do it very, very carefully. The very rationale which is required should be used. You should be able, because you have to think that whatever things are required for the interpretation of the large data, accordingly you have to put it. And whatever things are required for the modeling also, you have to think of it. Because modeling, when you see the ground level concentration, simply telling that, okay, this is the emission which we are thinking of it, and this will be the ground level concentration. You have to say what will be what are the ground level concentration that is expected near to the nearby hospitals there. What is the GLC that is expected near to the primary schools are there, or what are the GLC conditions that you are expecting to the sensitive area there. It is not done in this modeling. You have to do. In order to know that, you have to say, you have to be cautious in selecting the monitoring location for air sampling. That means, while selecting the monitoring location of the air sampling, you have to think how your report has to be, how your report should look like to be. What are the things your report should mention, must mention, you have to think of it. Then only you have to fix the monitoring location of air. Actually, the required monitoring locations are told like this. But nobody takes care of, and the majority of the people don't know. See, Suspended particulate matter, I have told that high volume, and SO2 I have told the bubbler and NO2 bubbler. Carbon monoxide oxidant. See, based on the population of the evaluation area, how many monitoring stations are there? But usually we put at 8, costly 8. Okay, fine. But see, your 2 points are there. See, I have mentioned suspended particulate matters, SO2, sorry, SPM, high volume, then SO2 bubbler and NO2 bubbler. Why? Because earlier high volume samplers were used for the SPM and SO2 and NO2. But nowadays, at least for NO2, it should be the bubbler mechanism, only sampling should be done. I haven't seen any of the laboratory having this facility for doing the NO2 sampling by bubbling method. I didn't see. If it is so, that is very good. You tell the laboratories when you are selecting at least what is your method of sampling for the NO2. It is a bubbling method, you see, because it will give more accurate results than the high volume. That is it. So this is for your information only. But it is not practical always. It is given in the IS 5182 part 14 2000. Recommended minimum number of stations population. It is a minimum. So it may not be practicable always, but if possible, you think of it. If it is okay, that is fine. And in your report also, you can say if the sampling station that you have selected comes within these standards, you can add one more point in your air quality report that based on this we have selected, this is the standard given, we have selected this one. It will give added advantage of your report. Then secondary data. For later interpretation, we may require secondary data. What is secondary data? What is it? Windrose diagram. See the Windrose diagram. I'm telling you first, you see the Windrose diagram. In the case of Windrose diagram, secondary data taking a single data from the IMD or something, or for the 10-year single Windrose diagram will not do. You try to make Windrose diagram at least 4 or 5 for your location expertise for 4 to 5 days. But one should know what is a Windrose diagram. Putting a software and making a Windrose diagram, I am not recommending. You can do. But first I am telling all of you try to learn how to draw a Windrose diagram. What is it physically? Manually. Learn to draw a Windrose diagram manually. First you learn, then use the software. That means even if you don't have the software, you can draw a Windrose diagram. You will have the velocity and the direction, etc. So how can I take the velocity and this direction? This it is very simple. If people are going for the field work, you take your kerchief. क्या बोलेगा? Handkerchief. हिंदी में क्या बोले? Kuman. Okay. You take the kerchief and keep it. It will show some direction that the wind is going. Then you take put a paper, a small piece of paper, you will send it. This from this to this, how much time it took it, you will get the velocity, finished. So 2-3 times in your location, you mark it in your notebook. It is very simple. Nobody say that it is you should use a meter only. If you put it monitoring this what is it, meteorological meter, it is good. You will get the velocity and this direction, everything. You will get it for 24 hours. But simple. At least if you don't have, we can do that. Then come and draw the Windrose diagram. How the Windrose diagram is to be drawn? I think it will require one another session for it. How to draw a Windrose diagram? Everyone should learn. That is, do it manually. That means at the center you can see what is there. At the center you can see what is there. At the center you can see, here you can see, at the center there is a surface and there is a round. It is told that it is zero. See, it is at the center, it is there, no? What is that? It is a time period where it was idle. It was calm. How much period it was calm, and it depends on it. It decides the size of the circle. Then see the wind where it. This is the direction. That means where it comes from this direction, or it goes to this direction. You have to know the highest. That is the name of a wind is given from the direction it comes, not to the goes. So see the monsoon, southwest monsoon and northeast monsoon. In June, July, it starts the southwest monsoon. And second comes the retrieval of monsoon that comes back, that is the northeast monsoon. That happens in the October, November months. That is the monsoon that we, 2 monsoons that we get in our country. So how to draw a windrows diagram if you know? Draw manually, you try it. It is very easy and it is very interesting. Just like you are drawing graph also. See there it will define these colors also, how you give, how much percentage you give, this one. You learn this one. If you learn this one, then you will become an expert. You can expert means you will be happy in drawing so many numerous Windows diagram. Nobody has told that you should do with the software. Manually you try, manually you learn, then only you can counter-check with the software. So try to learn this one. It will get one more session I think separately for Windows tags. Then so taking this one and the interpretation secondary data is required. Then another one, see this stack. In this stack also. In this stack also, I have seen the airport reports many times. I have seen gone through it. See, you know the wind pattern of a particular area. If there is an industry, you know it will be having a stack. And you know how to calculate the stack height also. When you calculate the stack height, you know what will be the shape of the stack also. Shape of the stack also will be different, no? So the stack height, how much it will be there? The wind, what is the wind direction? Then windrows diagram you will get it. Then you should be able to say what will be the. You should be able to say what will be the shape of the plume. This is plume. You know plume? See, plume behavior. In any of the airport reports, I haven't seen anything about the telling about the plume behavior. See, you should know as an airport expert, you should be able to say the stack height being this one and the wind direction and the wind pattern being in that area is this one, and usually these times the wind discipline behavior will be like this. You should be able to say none of the EIA reports I have seen. So you should be able to see, take the secondary data and try to describe on the plume behavior. It will give the added advantage of the quality of your EIA reports. As an air quality expert, suppose you are doing this one, you are putting in this report, see how you will also think, you will also be happy about your report. So make it a comparative report. Try to say the plume behavior, what is expected from you. See, this is I have given an example only. Then sometimes the plumes will go like this in a zigzag manner, or sometimes it may go there only. See, I have shown another photo now. See here how the plume goes. See here it goes in this way. See here it goes this way. See here also it goes this way. See, this is a plume goes on. So here in this case also you can see I didn't get the it is color is not visible. So now it is not possible. See, there are different plume behavior. You have to learn about the plume behavior as an air quality expert. You should be able to say what will be the plume behavior in this area and at what time. So the inversion when it will happen. See these calculations you have to say. See the wind direction. Then actual point source is there. Time average plume center line. See time average plume boundary. These things you have to do and do the calculations and make your air quality report a best one. Nobody can copy it. So better you do this one. So these secondary data is required. That is what is required. Meteorology, temperature required. Then relative humidity is required. What is the precipitation, wind speed direction. See, when you go for the air quality monitoring and air quality monitoring, people should give the real data that at the time of sampling, what was the condition, whether it was rainy or windy or sunny or dusky, they have to give. Nobody is giving. And what time the sampling has done, it is daytime or night time, it is also given. But what is the ambient temperature, it is not given. So the ambient temperature is required. And here you know when the stack comes, you know in that industry, the purpose industry, what will be stack temperature also. So the flame behavior, whatever it will be, you know. So this tells you can say. So try to learn drawing the Windrose diagram manually first. Second thing, try to say what shall be the plume behavior in that area if the industry comes at whatever times, and try to say if that plume behavior, what will be the impacts that is caused on the atmosphere. That also you try to say. Then your air quality report will become very excellent. Anything you want to ask, madam? Anybody? Anybody want to ask anything?
No, sir.
Can I continue?
Yes, sir.
Shall I continue?
Yes, sir.
Okay. See, then some more points to be remembered. What is that? A network of 8 ambient air sampling locations is occurred within a proposed voice setup, 10 kilometer radius to generate representative ambient air quality data. This is what we are doing. We are taking this point, minimum this 8 is required, but I have told what are the number of locations you have to take earlier. So this point we are sticking on. That a large flat space elevator at least 2 to 3 meters is needed to place instruments. Where a flat space elevator at least 2 to 3 meters is needed to place instruments. A high voltage sampler, you are going for taking the ambient air sampling. So 2 to 3 meter elevation is required. Then site should be away from major pollution sources. The distance depends upon the source, its height and its emissions. Suppose an industry is there, its stack emission is there, nearby to it, don't take an instrument to your first floor or the terrace of the first floor and collect the ambient air if there is an emission source nearby. Then site should be available for a long period of time. Why? Because I have taken the baseline data. Then later on also after the project implement also, suppose I want to know the status of the ambient air quality, that site should be available. So easy access to the site should be there anytime throughout the year. Then site sheltering and facilities such as electricity, water should be available. That you will ensure. This is another very important point. Height of the inlet must be 3 to 10 meters above the ground level. So inlet point of the there is a high voltage sampler. Now, is a high voltage sampler. I will show you. See, the high voltage sampler will be there now. Like say you are putting the high voltage sampler. The inlet of the high voltage sampler, inlet, there will be a inlet now. You can see. See, can you see this one? This is a high voltage sampler you are putting usually now. Can you see this one? What I am trying?
Yes, sir.
Okay. See, there will be an inlet. For this high voltage sampler, there will be an inlet. It will be kept like this. Okay. This inlet, about I am telling this inlet, it should be height of the inlet must be 3 to 10 meters above the ground level. That means that in a room usually it comes. So 3 to 10 meters above ground level it should be there. You should not keep it that much down. You should ensure. Some photographs I have seen, ambient air sampling photographs. It does not do like this, see like this. So at least when you send some photographs or when you put some photographs in the EA report, you ensure these guidelines. It ensures these guidelines. Otherwise, don't put it. Tell them to give another photo. Then collocated samplets 2 meters apart. See, if you are putting 2 samplets together, at least 2 meter distance should be there. Another sampler, you have to keep it 2 meter distance at least. So this 2 meter distance, this should be minimum 2 meter. I have seen photographs where 2-3 instruments are kept and sent a photo. If you get this photo like that, don't put it, because if you do not know this condition, then you will take that photo what the people are sending. If you know the condition, you have to send them back and ask them to give correct. This photo cannot be accepted because minimum 2 meter is required. Got it? So that be careful of it. Then there should be no nearby furnace or incinerator. That you may see. Then pamphlets are to be kept at a distance of 200 meters from unpaved roads and streets. The unpaved roads, if it is unpaved road, I have seen pamphlets kept near to a building in the terrace, near to unpaved road. 200-meter minimum, because if it is an unpaved road, because of the road traffic, heavy dust will come, particulate matter will come. These particulate matter will clog the pores of the filter paper, so you will not get the accurate answer, accurate report. So keep at minimum 200 meters from the unpaved roads. Don't take a photograph where the unpaved road is seen and this instrument is seen. Then be careful of it. Okay. Then certain trees may also be source of PM in the form of detritus, pollen, and insect parts. Certain trees. If you go near to the cement industries, usually you can see that. Or if you go to the mining industries, if the granite mining or something is going on, the trees will have would have been covered with the dust, everything. So when the high voltage sampler you are putting it near to that tree and sucking the air, then what will happen? The dust from these leaves will also fall in it, and it will answer for the particulate matter. So this can be avoided by locating samplers by placing them greater than 20 meters from nearby trees. I have seen so many photographs where the air samplers are kept and heavily trees are there on one side. So at least take a photograph avoiding that even if you are kept there, or at least don't put such photographs in your ear. So these points keep in your mind very carefully and examine every photo that you are going to put in the EIR report with regard to air pollution monitoring, ambient air monitoring. You keep these points, check with it. If it is okay, you put it. Otherwise, don't put it. You ask the party to send another photo. Then site should be 1 meter away from observing such as observing building material. Then another point that is to be considered. See, we are measuring the PM10 and PM2.5. See, what are the different relationship between these different size ranges of ambient particulate matter? See, there is ultra fine particulate matter, PM, we can say 0.1. Accumulation particles are there, that is PM 2.5. Then other is there, coarse particles are PM10, again are there, these suspended particles. See here, in the PM 0.1, the carbon, sulfuric acid, condensed metal vapors, etc. will come. In 2.5 condensation mode and droplet mode, you can see sulfate, nitrate, ammonium, organic carbon, elemental carbon, heavy metal, fine geological, but these are the accumulated one. Then comes the geological material, pollen, sea salt, etc. in PM10. Why we are measuring PM 2.5? Because this particulate matter is important because it is likely to affect our respiratory system. So if we say that the PM10 is high, very high, you can make people panic. If you say there is no PM10, somebody may say that your result is wrong. You are providing false materials. So what is the actual content you have to tell? That is important, the PM10 and PM2.5. So PM10 and PM2.5, these are the particulate matter where the aerodynamic diameter, particle aerodynamic diameter. So aerodynamic diameter is 10 and another is 2.5. What is this? These are the particles that may go into the nostrils of respiratory tract of human. See, in our body, how the particulate matter goes through the nostrils. In the nostrils, we have two openings. The nostrils have two openings. These nostrils are made of mucus layer, and mucus layer we have hairs, and because of this mucus layer and because of these hairs, the particles in the size of PM10 will be caught up there. That means even though the particles of PM10 size goes into our nostrils, because of having the mucus layer, what is it? Mucus layer is what is it, moisturized. The moisturized mucus layer and with air, these PM10 particles will be blocked there. But even smaller things, that is the even smaller things, that is the fine particles, ultrafine something, PM2.5 and ultrafine also, it goes into the nostrils, then it reaches the alveoli. In the alveoli, it reaches the alveoli means in the alveoli, there happens the actual transport of what is the exchange of gases between carbon dioxide or the oxygen or what is exchange. We inhale air, and the air goes to the alveoli. In the alveoli, in the alveoli, the small blood vessels. What is that name? Small blood vessels, minor blood vessels. I don't remember that name. In the alveoli, in the exchange of gases happens. That is in small minute vessels. Okay, leave it. So in the alveoli, the exchange of gases happens. How the exchange of gases happens? Yes, it is in the capillaries. Minute blood vessels are called the capillaries. In the capillaries, the exchange of gases happens. That is the air that has come into the alveoli in the capillaries is rich of oxygen. Then the oxygen will combine with the hemoglobin in our blood, in the capillaries. So suppose the air we are inhaling is having this PM2.5 particles, it will clog the capillaries and it will clog the alveoli also. So in the capillaries, then if it is carbon monoxide going on, the hemoglobin what is having? Hemoglobin has got greater affinity to carbon monoxide than oxygen. So if carbon monoxide that we have inhaled is there, then hemoglobin will readily combine with the carbon monoxide than the oxygen. So instead of forming oxyhemoglobin, carbonohemoglobin will be formed, which is a stable compound. Because the oxyhemoglobin is unstable, then the capillaries take this to the tissues. There they take the carbon monoxide and give oxygen to the tissues, thereby forming ATP, NADP, etc. No, ATP, NADP, H2, etc. Then coming back, they will release this one and form the ADP, AMP, then NADP, etc. etc. So that's the possible observation. So here the PM2.5, if it goes to the alveoli and close the alveoli, then this in the capillaries, this exchange of gas will not happen. It will lead to a condition called asphyxia, anoxia, etc. and lead to death also. It may become fatal. That is why we are measuring the PM2.5. So if we have to get the accurate value of PM2.5, PM10 in a particular area, these points to be taken care of. Where to keep these monitoring stations? Then ambient air pollutants and their standard measurement procedure. We have seen. Once I saw once I don't remember where it is. There I saw a person is monitoring the ambient air.
Hi, sir, we have a query. Can you please go back to the previous slide?
Previous slide, okay.
Sir, what is the difference between concentration of PM10 and 2.5? When relation?
Their relation.
Okay. That's very good. I think I'll be coming later on. Anyway, I will tell you. Actually, there is no relationship. We cannot say these fine particles shall be this much percentage of these quartz particles. Can you say? It's not possible. But
We are being told usually it is taken as 60,
No, I will tell you.
40%,
Wait.
So I don't think that. Can you please elaborate on?
Wait 1 second. The relationship you cannot say because you cannot say that fine particles will be this much percentage of the quartz particles. But usually PM2.5 will be less than PM10. That only we can say. No relation can be said because PM10 will be answering including PM2.5. But PM2.5 will not be answering including PM10. So PM2.5 will be always lesser than PM10. But percentage wise we cannot say. Anything else you can ask me now? Yes? Anything else?
No, sir.
Graphs
Okay.
में आने के
Fine.
Sir,
So keep in mind, PM 2.5 will always be less than PM 10. If PM 10 is PM 2.5 is greater than PM 10, that result is what? Because all the PM 10 will answer with the PM 2.5 only. But all the PM 2.5 will not answer with the PM 10. So PM 2.5 will always be less than PM 10, but we cannot say any ratio. Because if I take some dust in my hand, I can never say that the fine particle will be this much percentage of the coarse particle. Can I say? It's not possible. Even you take, what is it, puttu podi, or you take dosa podi, or idli podi, you cannot say that fine particle will be this much percentage of coarse particle. Am I right? Am I right? Am I right?
Yes, sir.
Okay. Yes. See. Can you hear me?
Yes, sir.
Yes, sir.
Okay. Shall I continue? Shall I continue?
Yes, sir. Yes, sir.
आपको बोरिंग होता है तो बोलना पड़ेगा. Ambient air pollutants and their standard measurement procedure. See, once I have seen somebody doing the ambient air quality monitoring, I went to the station and I saw it. He came there, he switched on the high volume sampler. Bas. Then he put a filter paper. I asked him, how you put the filter paper? So I simply put the filter paper. Do you know that it have two sides? Okay, sir, two sides. How will you put it? I will put it where the number is there, other side I will put it down. Okay, finish. Then in the side where is the impeller, it is there. What is there? Water only. Why you put water? Sir, we put water for 8 hours and take the sample and send it to the water. See, you will not get anything. See, here I have told this. Equipment, high volume sampler, SPM, RPM, so many things I have seen. This is pollutant. See, it is impinged sulfur dioxide. It is done by improved Western Geek method. That is it. Absorbent media should be there. So that is STCM. Sodium tetrachloromercurate is the absorbent media. So the imbinger with the sodium tetrachloromercurate should be there, 30 ml. So he has not kept there, he has put water. But NOx, see, NOx, it is Jacob Hoshizer method. Here the NaOH is the solution, and this distillate can also do here. Okay, so the medium which is required, absorbing media which is required to be kept there, it is depending on the analytical method. So where the analysis is, when the sampling is done, these things should be taken care of. Second thing, I asked him, how you adjust the flow rate? Then how you know that your sampling period? All these things I asked, then he was not knowing. Because flow rate, what is the flow rate? See, if it is 0.8 into 1.2 m³ per minute, that is the flow rate here for the SPM. Then for SO2 and NO2, it is 0.5 LPM. See, if you take a single high volume sampler for both these integrated for SO2, SPM and NOx, it is not acceptable nowadays. Earlier it was there. In 1984-85 it was acceptable combined, but now it is not acceptable because the flow rate for this SPM, PM 2.2 and PM 10 is different, 0.8 to 1.2. And for SPM, SO2 and NOx, it is 0.5 LPM. See, you know the 0.5 LPM or 0.8 to 1.2. So you have to note down, you have to adjust the LPM. Usually they do with the 1 LPM. Why 1 LPM it is done? It is easy for calculation. See, 1 LPM, that is 1 liter per minute. 8 hours he is doing the sampling. That means how much volume sample, how much volume air he has taken. That is 1 liter per minute. That means 1 hour, 60 liter has come. So in 8 hours, 480 liters has come. So the SO2, what is there in this one, from the laboratory, this much microgram is there inside this impeller. So in this impeller, it is for the 480 liter. So why microgram sulfur dioxide in 480 480 hours? No. Yes, no, 480 liters of air. 480 liters of air. 480 liters of air means then he have to he have to calculate by m cube. Then only the calculation they can give for this much microgram per m cube. So if the laboratory party has to report microgram per m cube, the laboratory person has to know which flow rate this man has taken the sampling. Secondly, how much time they have taken the sampling. There is a timer in the high voltage sampler. The timer reading, initial reading and final reading only will give you the time. It may not be 8 hours. It may sometimes 7.62 hours or 7.92 hours or 8.13 hours. So if they have the timer readings also, actual time that has been worked. Suppose he has put in early morning in 6 o'clock. And 2 o'clock he is coming, he is telling 8 hours is over. In between 1 घंटा बिजली नहीं था, मतलब 1 घंटा चले गया, 7 घंटा ही सैंपल हो गया, लेकिन ये टाइमर तो बोल सकता है, लेकिन ये आप को बोलने को नहीं हो सकता क्योंकि ये उधर नहीं था, इसलिए टाइमर भी चाहिए, टाइमर का रीडिंग भी चाहिए. So see when you ask these people, they are giving the results, you can ask the field data sheet also. There once I went there and once I saw that person, he is not knowing anything about the what is it, timer. He is not knowing anything about the flow rate adjustment. He is not knowing about the what is it, media, absorbent media, which is to be used for each gas. That is the. I asked him what is your qualification. He said he is MSc in environment science. Okay. So I told him you will learn well and come next time and do sampling. Please don't. At least you can say you don't know. Don't say these blunder things. So keep in mind. My dear friends, you keep this in mind. You ask this air monitoring people what they are doing actually. Okay. So this table you can take. You will be getting copy of this slide. So this thing you can take it from that slide. Keep it. The emission inventory. Emission inventory is required for interpretation of the data because in your report you will be putting the interpretation. Data interpretation you have to do. So their emission inventory is okay. I have seen a lot of people discussing emission. What is emission? How I can get the emission? What is emission? So many people discussing. In Gaurang also I have heard. It is not fair. You should know what is emission. Emission inventory is a major tool for identifying the sources of pollution and the quantitative expression of pollution load in a defined area. Because initially I told you there may be cumulative effects. So while selecting this ambient station monitoring point, the cumulative effect you have to get. So emission inventory is okay. Emission inventory from where you will get, you can see, you find out. Then study would involve preparation of detailed emission inventory with the estimation of emissions from various activities such as. You always think that when we say emission details, people always think that, oh, there is a stack in the nearby industry. From that industry only you have to take emission. No. See, that also you have to take, but detailed emission inventory means it is such as vehicular emission, industrial emission, residential emission, commercial emission. These to be made using primary as well as secondary data through questionnaire, survey, wherever necessary. See, the socio-economics people are doing the questionnaire survey, or they are having the what is it, cluster group meetings or something. In that cluster group meeting also, these are to be included in the socio-economic survey. This will be used for the air quality experts. But in the focus group discussion, yes, focus group discussions, socio-economics is doing. But in their questionnaire also you can add this one. The air quality expert, you discuss with the socio-economics people also. That is, these experts should have interaction between each other. Air quality expert has to say to the socio-economic expert, hello जी, मेरे को उधर इतना डिटेल चाहिए, आप उधर फोकस ग्रुप डिस्कशन चलता है ना, नहीं तो क्वेश्चन का कलेक्ट करता है ना, उसे ये 2-3 क्वेश्चन भी उसमें डालो, मेरे को आंसर दे दो. See, air quality expert only knows it. Socio-economics वाला नहीं जाता है. Air quality expert उसको बोलना तो वो पक्का करेगा. See, don't depend on socioeconomic expert only. Even though who is going for this biodiversity or who is going for the water or for the other also, you can see who also keep on this. So this session everybody is hearing now. So any expert can keep this in mind. You can say, see, you go and tell the airport expert, here this. So emission details does not mean that only the industrial stack emission. Then impact of pollution from these sources depends on many factors. What are they? Vicinity of emission sources, concentration of pollutants, temporal and spatial variation in emission pattern, and receptor types. But suppose in the nearby industry you are not getting any data, you can have some research papers, you will get some second data for the similar industries, etc. Then eccentric survey and primary data generation is zona influence. Around each monitoring location will be a major component, that is 2 into 2, 4 square kilometer. Other cases we are doing the buffer area, that means 10 kilometer, but extensive survey is required for this interpretation of air quality data for 4 square kilometer. Keep it in mind, because it is a major concern about the emission. So emission inventory should have point sources, area sources, and line sources. In part 2, this is a part 1 session I am taking. In part 2 session data interpretation, I will come to point sources, area sources, and line sources. Many people are discussing here point source, area source, line source, but in majority of the reports, I have not seen this one. Okay, drilling, but it is not seen. You keep trying to learn and incorporate in your report. Then minimum distance of sampler from road. If you have a traffic road, what is the traffic distance? You have to see average traffic vehicle per day. You have to take a count of vehicle per day. So take the count, you can see that minimum distance of sampler from the road. See, if it is less than 10,000, greater than or equal to 10. So if the vehicle, as the vehicle increases, see 20,000 becomes 30 meter. So as the number of vehicles increases, it goes, and the distance also increases. See, another point I have given down, lower detection limit. Pollutant SO2 and NO2. Lower detection limit I have given. See, this is important because, see, here also I have given, I think so, minimum detection limit. So here you see, SO2 and NOx minimum detection is 0.04 microgram per MQ and 0.03 microgram per MQ is the NOx and SO2. Then PM 2.5 is 0.5 microgram per MQ. So I have seen results less than this. How it is comes? Below 0.04 microgram per MQ. If the improved Western Git method is there, the result is wrong. Because the minimum detection limit is that one. So when you go on checking the test reports for interpretation, you see this one also is ensured. Then ambient air sampling. This is what you have seen this one. High voltage sampler, this is the most popular method. Non-volume of air is increased, the sucked and increase in weight due to trapped particles. That's it. Gravimetry analysis only, PM levels is in microgram per MQ. The stack emission monitoring. See, this is the stack. See the plume behavior. See the plume behavior. This is the stack. You see, I think the courtesy of this photograph goes to Denjit, I think. Oh, somebody has taken. Oh, if it is not so, maybe for Siddhartha or somebody. Okay, see the plume behavior, how it goes. This one goes like this. This goes like this and comes down. See, this goes straight and then widens. See, so these are the things you have to see. Okay, see, this is the thing. This goes like this and goes this, comes down. Again goes. This straight goes and widens. See, the plume behavior, you have to be able to say the plume behavior. So this stack emission is measured by stack sampling kit. It is carried out by diverting a part of the gas stream to the sampling train. And the sampling of the stack monitoring is not very easy. It is very complicated. And the stack monitoring kits conserve so many things. See, nozzle sampling, particular characters, so things. Now I'm not going to details of it. See, item, stack monitoring, specification of stack monitoring equipment. You can go through this. That is simple. When you get this slide, you can go through it. Copy of the slide, you will get it. See, these points I want to specifically say. This sampling is done at some levels. Stack sampling. See, some people are standing here. This is the platform for sampling. That is, sampling site should be located at least 8 stack diameters downstream and 2 stack diameters upstream from any source of load system such as bends, fittings, etc. See, if the stack is having, sometimes they may be having some bending straight initially and then goes up. So after leaving this bending, from below minimum 2 stack diameters, that is, take the diameter of the stack, minimum 2, if the diameter is 5 centimeter, then 10 centimeter. No, 2 meter, it is 5 meter, then it is 10 meter. From the above 8 into stack diameter. So if it is 5 meter, then 8 into, that is 8 into 8 into 5, 40 meters. So in that way, you have to have a sampling point. And the stack will be having a porthole. A porthole. Then sampling points must be done at a number of traverse points. They see, I have shown this one. This one. These things will go into the stack. So you have to have the traverse points and measure it. Then only stack emission can be done. How many number of traverse points you have to take, it is given, and the minimum number of traverse points, the calculation is given. Then the calculation of the emission is also, flue gas analyzer is also required, because flue gas analyzer is required because oxygen concentration you have to take it, that is from the atmosphere. So this, you go through this one, then come to the calculation of emission rate. If the emission rate you can calculate from this. So if you have to calculate emission, if you say new industry, you know the stack rate what will be, you will come from the calculations. You will be knowing what will be the emission or what will be the velocity of this stack, and you will find out the isokinetic velocity of the monitoring purpose of the emission gas, and from there you will calculate the emission rate also. So then you will have the emission. This is what I have told. Okay, fine. Coming to the soil sampling. Soil sampling is to collect a soil sample that is representative of that one, to obtain information about a field and information to make management decision about the nutrient management, soil problems management. That means when you give an interpretation of the soil result, you have to say the nutrient about the nutrient management and also soil problem management, where it is agriculture field or whether it is used for the other purposes or for the industry or for the curriculum purposes or something else or making bricks or something. You have to say soil problem management. That is why we are doing the soil analysis. In any of the report, I haven't seen the soil quality interpretation about the nutrient management or soil problems. Try to include this one. See, sampling a field. Two basic methods are, one is the random and the zone methods. What is the random sample? This is the random sample in a field. Then what is the zone method? This is the zone method. See, very simple. How to select the suitable sampling time? What is the time? See, when I give the tick mark itself, it is very clear. The first one is okay. This one is okay. This one is okay. When you see, take the sand in your hand and see, then this one is not okay. See, when you put your leg on it, this is not okay. This is also not okay. Okay. Then, see, taking a representative composite sample. How? Non-representative composite sample. If you take this one, this one, this one, this one, no. But sampling complex samples, I mean non-homogeneous field. This way you can not take, this way you can take. See, this one, here also. Then representative complex sample, homogeneous field. This is enough. Then sampling tools. Soil tools, you know, agricultural soil, or what? Or the soil agar. This is the commonly used one. This also you can use it. Shovel is also a suitable go. Okay. Then taking sample at a plow depth. Plow depth means when you plow this one, that is called the plow depth. There is a plowing cattle, it's a plow depth, 15 to 20 centimeter. Soil agar with this in, spade you can use it. Mostly this one is used. Then collecting soil samples, scrap the soil samples from the return before sampling. Collect sample from the surface layer using the this board mentioned this none. See any of these you can use it. Then take the sample and discard this part, you take these two. Save, that is. From the figure itself, I think it is very clear. Mix thoroughly, like fragments. Put it in a polythene bag and send it to the laboratory. So these sample preparation, collect composite samples. Old spots should be sampled separately. Transfer sample to plastic carry bags like this. Record the sampling location of the GPS coordinates. Information read on the lab use. Okay. So what we have covered with this, my time is running over. So learning new or refreshing knowledge. I don't know whether
So
you
for
are
soil
learning
sampling
new
is
or whether you are refreshing your knowledge. And what we have covered is these things. Main factors that may affect the
Nair, sir.
precision and accuracy of the result. Then
Nair, sir.
in the case of the sampling, emission inventory, then emission monitoring, how the soil environment is required, what should be covered in the interpretation of soil samples. So these are things I have
Nair,
covered.
sir.
See, you see, if you don't know, ask. You will be a fool for the moment, but a wise
Nair,
man
sir.
for the rest of your life. So you can ask. Okay?
Sir.
So thank you. You can ask. Okay. Now you have enough time to ask me anything.
Now ask.
सर सॉइल सैम्पलिंग में जो सैंपल की डेप्थ होती है 30, 60, 90 होती है 15 to 30 होती है सेंटीमीटर.
In most of our reports, we are giving it as 30, 60 and 90 centimeters. The sample is collected from different depths of 30, 60 and 90 centimeters.
30, 60,
Is that
90,
correct?
30, 60, 90 सेंटीमीटर नहीं चाहिए यही बस. क्योंकि ये टॉप साइड निकाल के उसके नीचे आता है. टॉप साइड निकलता है.
जो टॉप सॉइल होती है उसको हटा के फिर 15 to 20 लेते हैं.
टॉप साइड
एक सेंटीमीटर.
Before the top side, sometimes overburden may come, but testing the overburden is not required. But near the top, remove the top side, and the overburden is not required, so no need to go to that much. That is why it is called a 0 to 50 centimeter, because agriculture of us, that is it. Flowing depth. This is it. Flow depth. PLOUGH, that is the correct word. Spelling mistake. Okay.
सर कह रहे हैं टॉप सॉइल को हटा के 15 से 20 के आसपास.
Anything else?
Okay, sir. Anything?
Sir, is there any chances of getting PM 2.5 more than PM 10?
Getting PM 2.5 more than PM 10, no chance. If it is so, anything wrong in the gravimetry analysis, because see, once the filter paper is coming from the field, it may have some moisture content. After it reaches in the laboratory, it is to be desiccated, that is removal of moisture, then taken the final weight. But some people either do with hot air oven, put it, or without putting it, they take the weight. So the gravimetric analysis mistakes, PM10 may be less than PM 2.5. In such case only it would come. But in practical, PM 2.5 can never be greater than PM 10. Because the sampling for both these are done simultaneously for the same time. And PM 10 always answers with the PM 2.5 also.
There are specific conditions.
Okay.
But sir,
Discussion करते हैं. बताइए.
Yes.
चलने दो सर.
नहीं.
मैं बताऊं. कुछ conditions हैं उन cases में. But sir, there are certain conditions. I just not remember, but there are certain conditions where PM 2.5 is more than PM 10, either equal or more than PM 10.
No. PM 2.5 more than PM 10. See, suppose it is an what is untarred road, and not bituminous road or the normal village road, then the vehicles are passing. Then you are telling that PM 2.5 may be greater than PM 10. what is PM10? See, the answer for the PM10. The PM10 is collected in a filter paper. Am I right?
Yes, sir.
Yes. PM10, how it collects PM10, the pore size is greater than PM2.5. Am I right? So all
Okay,
the
sir.
PM2.5 particles will be collected in PM10 also. But the PM10 particles will not be collected in PM2.5. That's the basic principle. So if any result has come there, it is wrong and there is no chance.
Okay, sir.
Because the particles are collected in this filter paper because of the difference in pore size. PM10 filter paper collects the PM10 and PM2.5 both. But PM2.5 filter paper will collect only the PM2.5. So the gravimetrically and scientifically, the PM2.5 cannot be. But if it is a different time you are taking, suppose morning time PM2.5 is collected and another time PM10 is collected, then there is likely chances. But here PM2.5 and PM10 they are collecting at the simultaneous stage. If it is so, it will not. It will never.
Okay, sir. Right.
Okay.
Sir. Any
Okay,
other questions?
Nitin?
Thank you, sir. Sir,
Sir, one more question, sir, please.
Right?
How much quantity is minimum quantity is required for the sampling of groundwater and surface water or soil sampling?
Usually it is 2 liters. If only
2 liters.
chemical parameters are required, it is only 2 liters.
For
If
groundwater and
toxicity is
soil
required, it will be
sample.
required 10 liters, 10 to 15 liters.
And for soil sampling, sir?
For example, what can I say? Kilogram wise I cannot say. I can show my hand
Okay.
only, this much is enough.
2 kg, maybe 2 kg, sir.
2 kg, अरे कैसे 2 kg हो सकता है? ये पार्टिकल्स हेवी है तो 2 kg भी मेरा एक ही हाथ में आएगा. लेकिन दूसरा क्ले पार्टिकल सब ये है तो 2 kg तो मेरा 2 हाथ में लेना पड़ेगा, इसलिए हाथ से इसलिए केजी बोलने का नहीं होता है. लेकिन क्या है, जितना आपको देख सकता है जितना चाहिए ऐसा ही हो सकता है. एक ऐसा बना A4 से इसका ये है ना, A4 से इसका एक सिपलॉक A4 से इसका सिपलॉक कवर लेते तो उसमें कितना आएगा उतना ही बस.
बच्चों वाला क्वेश्चन
मतलब क्या है, उधर whatever testing to be done, one is the pH and the moisture content, then water holding capacity, and the what is ion exchange capacity, then available nitrogen, available phosphorus, total organic carbon, then I think this much only. Anyway, so only for this what is water holding capacity, they cannot try and take that 10 gram sample and put it like that only. Then other things they will give the air trying. After air trying, they will separate it with fine particles and grind it and then sieve it and then take the sample 1 gram, 2 gram, 5 grams. That is enough. So maximum quantity of all the parameters of the analysis, it may require maximum 10 to 15 grams. That's enough. So what we cannot send 10 to 15 grams, no?
Yes, sir.
That's right. So this much A4 says this Ziploc cover, how much quantity comes, that is enough. That will be. Because heavy I cannot say, because the what is it, 1 centimeter, 1 square centimeter medium एक आइटम 1 किलो होते तो दूसरा आइटम 2 किलो होगा, इसलिए वही तो डिफरेंट होगा ना. मॉइस्चर कंटेंट में चेंज होता रहता है, इसलिए किलो मेजरमेंट मेरे को बोलने का नहीं होता है, इतना ही. ओके.
Yes, sir.
Sir, I have one more question, sir.
Yes.
Sir, when composite sampling is required, sir, in case of water.
Okay. Usually we for the gram sampling, na. Composite
Yes, sir.
sample is okay. See, suppose you are taking a water body. If you are taking a stagnant water body and you feel, you take the pond, in the case of a pond. If you take the pond and you want to take from different places, if a small pond is there, I'll show you. See, if it is a river,
नहीं होगा वो स्क्रीन से अच्छा अपन ने कर रखी
okay, this you can see. If it is a river,
Sir,
at
one
least this,
minute,
This
sir,
is
1 minute.
having minimum mixing point. You can take this. This is enough. 1 gram sample will be enough. Or you feel that upstream, midstream, downstream, etc. You need. You can take a composition. Or in the case of a pond, suppose it is a pond. You can see this one. No? What I'm trying, you are
Sir.
seeing. So if it is a pond,
No, sir,
see,
not with.
here there is a road going. Here there is a road something going. You want to take a sample from here. You take another sample from here. It is stagnant now. You take one from here. Suppose you get a country board and you go inside, you can take another sample from. Take this and make this. So in such cases, better to put it composite. In such cases. Then river also. If you feel that you care,
Industry waste water.
Integrated.
in the river also, if you want to see that some industry or some sewage effluent or something is coming, you want to take one sample from here, another one from here, or from here, then also composite. That depends on your discretion. Otherwise, not right. But in pond, if you take, that will be better. See, when I say about the river, one more point I want to say. 2-3 days back, I saw something about the discharge in the river. What's it? Volume. Water volume of a river or something. In the group, I saw some discussion. See, how you can calculate the discharge of this river. If you want to calculate discharge of a river, see, velocity meter, even if you don't have, you can simply calculate it. Suppose, see this one now. See, how is the river going? It is be going like this. This is one side land area, this is another land area. See, this will be going like this now. This depth you can calculate it. How much? It will go. You walk it, then you will get it. How much length, depth of your leg goes in. At least. Or you take एक लकड़ी लेके ऐसे जाते हैं तो भी हमको कितना पानी नीचे है इतना इधर. Okay. So you will get this much. Then you will get the speed of it. You put some this. If it is a way you are standing here, you put some paper here to reach here. How much time it takes? So the velocity you get it. The velocity and area of cross section. Velocity and area of cross section. You will get the discharge. Finish. That is a very simple calculation. Okay. Okay, fine. So that is about the composite sampling. Then anything else?
Integrated approach.
Okay, if you have any more doubts, now my time is over, 4:30 is over. I'm sorry for what is it? Boring लगा तो मेरा problem नहीं है, subject का problem है. ऐसा boring करने के लिए I'm sorry. But if you have any more questions, my time is okay. I can answer. Doesn't matter. Even if you later on feel that you forget to ask this question, you would have asked this question. You ask me. That doesn't matter. You can send me mail or by message. Remember always one thing. If you don't know, ask. You read once again or many times. If you don't know, ask. Because asking questions, you may be thinking, oh, it is a simple question. It may be a simple question or it may be a blunder question. Don't think like that. Because you will be a fool for the moment, maybe. But you will be a wise man for the rest of your life. So, go on asking questions. You ask questions. The rain does not ask any permission. It falls, it cleans us, it teaches us to keep going. So, you have made these kinds of great. See. So, you keep on asking questions. If you have, later on you think that you have forgotten to ask something, you can ask. Send mail or WhatsApp. Okay? Shall I stop?
Okay, sir.
Okay, thank you.
Thank you so much, sir.
Thank you. Here you can see air, water and soil.
Second part you was. The first part, second part.
Second part होगा जी.
विमल, इसका आंसर बता दे.
स्टैंडर्ड टेम्परेचर
क्या
प्रेशर
होती है स्टैंडर्ड टेम्परेचर?
25 डिग्री
25 और 1
और एक बार प्रेशर 1 ATM प्रेशर और
1
ATM हॉस्पिटल
सेकेंड
प्रेशर
पार्ट आज नहीं है, सेकेंड पार्ट बाद में आएगा. अच्छा आपको डेट दे देगा.
ठीक है.
25 डिग्री सेल्सियस टेम्परेचर एक बार प्रेशर ये अगर मेजर कर रहे हैं
If you feel good about this section, you tell. If you feel bad about this section, also you can tell.
वो पता नहीं ना.
ऐसे कहते.
Okay? Shall I leave? Okay, can I leave?
Yes, sir.
Yes, sir.
Thank you,
Okay,
sir.
thank you. Thank you for your patience.
Sir, please share your slides. Sir, this PPT slides.
Slides are given to HR, HR will share it.
Okay, sir.
Or if you want, if I can share in group, I can share it.
Thank you, sir.
Okay, sir.
Okay. Bye, thank you. आप लोग